Southern blotting. Southern blotting is used to detect the presence of a particular sequence of a DNA by using a molecular probe. So this is a technique for identification of a small piece of DNA or for the identification of a particular mutation by using a probe. This technique is called as Southern blotting. Southern blotting is named after its inventor, the British biologist Edwin Southern. He identified this technique in 1975. There are some other blotting techniques. Those techniques also use the same principle. That is the reason they are named in reference to the Edwin Southern's name. Here we can see that how these blotting techniques have been named. Here we can see that Southern blot is used for identification of the segment of the DNA or for the identification of particular polymorphism or DNA from the sequence of the DNA. On the other hand, Western blot, which is used for identification of particular protein in a mixture of the proteins. And the third type of the blotting technique is called as Northern blot. Northern blot is used to identify RNA from a mixture of RNAs. So, based on the name of its inventor, whose name was Southern, these techniques have been named. So, Southern blot is for DNA, Western blot is for the proteins, and Northern blot is for the RNA. Southern blot is a type of the molecular biology technique which is used to identify a particular sequence or a sequence of a gene. This technique can also be used for identification of the particular polymorphisms or a particular mutation from the sequence of DNA.